Hi, this is Anirban Mukherjee, and I will be talking about transport between nucleus and cytoplasm. Nuclear transport. Now, there are different proteins present within the cell which may or may not need to move inside the nucleus, right? So, how to determine which proteins actually need to go inside nucleus? There are several levels of control to it. Initially, the only way to get in or get out of nucleus is the nuclear pore complex. Also, there are other criteria like presence of karyopherins and nuclear localization signals and nuclear export signals. Karyopherins are proteins that carry the cargo protein in and out of nucleus. Essentially, they are of two types, that is importin and exportin. Nuclear localization signal is present on proteins which needed to be transported from cytoplasm into the nucleus. Importin binds to only those proteins which contain the nuclear localization signal and transport them from cytoplasm into the nucleus. And exportin, on the other hand, binds to only those proteins which contain nuclear export signal and transport them from the nucleus to the cytoplasm. Now let's look at the import mechanism. Let's just say there is a cargo protein containing the nuclear localization signal and there is a importin protein. Now importin binds to the cargo protein containing NLS. Now this whole complex together moves through the nuclear pore complex or NPC and moves inside the nucleus. Inside the nucleus there is a RAN GTP. When it binds to the importin, it allows the dissociation of the cargo protein. So the cargo protein gets released inside the nucleus. Now what will happen to the importin which is bound to RAN GTP? There is a phenomenon called RAN cycle. We have already seen that the importin is bound to RAN GTP, and this together moves through the nuclear pore complex and moves out into the cytoplasm. Once RAN GTP and importin has reached the cytoplasm, RAN GTP is converted to RAN GDP by RAN GAP. It's a GTPase activating protein, right? This conversion actually helps in dissociation of importin from RAN GTP, with importin being released outside the nucleus or inside the cytoplasm. Once RAN GDP is produced, it binds to another factor known as NTF2 or the nuclear transport factor. Together, RAN GDP and NTF2 is again transported back to the nucleus. Inside the nucleus, RAN GDP is again converted to RAN GTP by RAN GEF. GEF is a guanine nucleotide exchange factor which converts GDP to GTP. Once RAN GTP is restored, it is again ready to bind to importin and help to transport importin out of nucleus. This was the RAN cycle. Now export. Proteins which are present within the nucleus or certain molecules which are present within the nucleus may need to go outside, right? May need to go into the cytoplasm. Let's say there is a cargo protein containing nuclear export signal. To it, exportin and RAN GTP is bound. Together, they form a complex. This whole complex moves through the NPC out into the cytoplasm. Now there again, RAN GTP is converted into RAN GDP by RAN gap. This allows the dissociation of the cargo protein itself and the exportin. And after this process, again RAN GDP is again transported back into the nucleus by NTF2 factors. So that was all about import and export of proteins in and out of nucleus. So let me know if it was helpful, and thank you.